This is an introduction to the tutorial that I'll be doing on visualizing and running data analyses in Excel. The data that I'll be using was collected in the northeastern part of Pennsylvania in the glaciated Northeast, and it was collected in some forest stands located just north of a lake we call Lake Wallenpaupack. I also want to draw your attention to this other lake that's north of Lake Wallenpaupack called Lake Lackawanna. It is a glacial scour lake that you'll be using data from for your assignment this week. If you were to go and be at within these watersheds, you might see that the forest looks somewhat like this in some areas, particularly where there's a lot of water, maybe some seepage. But what we know about that area is that there are a lot of stressors on this forest environment. We have a very high level of deer browsing going on in the area. We have hemlock woolly adelgid, so it's decimating the woolly adelgid is decimating the hemlock population. And we have an increase in this invasive hay-scented fern. It is a native, but it is also invasive, and it's not edible. You can see it down here for the deer. So there are some interesting interactions between these stressors and their effects on the forest system. So part of our research is really to understand those effects of multiple stressors, the deer, the woolly adelgid, on carbon pools and fluxes in these forests. And some of the data that we're collecting is on vegetation, so thinking about the above ground biomass, the litter, so what's falling off of those plants, the soils and their soil properties, as well as the microbial communities that live within those soils. So soil microbial community composition as well as functioning. And we're going to be focused today on looking at respiration rates from soils. So these microbial communities and their ability to respire or break down carbon. So how did we do this study? Well, luckily there are two long-term deer exclosure areas in this forest. One is in an oak maple stand, the other is in a hemlock stand. That distinguishes between an area that would be affected by the woolly adelgid versus one that would not. Both of these exclosures were built in 1994 through 1995. And we've paired sampling within the exclosures with plots outside of each of these exclosures as well to get at deer browsing intensity over a fairly long time frame. And just to kind of give you a on-the-ground picture, we can see the oak maple inside the exclosure versus outside. We can see some visual differences there. And then we have the hemlock inside and outside, and they have some differences, but maybe not as many. And so there seems to be some differences inside and outside, but also between the forest types. And so that's the data that we'll be looking at today. You don't have to know too much about the sampling. Other than that, we are summarizing the data that we're sharing with you today is really a summation of a number of different samples within sampling plots in these 4 different regions. So here's the data set that I'll be sharing with you today, and I just want to go through the different columns to give you an understanding of the data set. We have the months, meaning that we collected data in June, July, as well as August. We collected that data in the hemlock areas as well as the oak maple areas. In each of those areas, we had exclosures and non-exclosures for each month. And this plot information is a specific plot within each of those exclosures that we sampled. And then we have our response variables, so what we actually measured. We measured the CO2 flux or the carbon dioxide fluxes. So thinking about the microbial communities as well as the root respiration rates, so how much carbon dioxide is coming off the soil. We measured the methane fluxes, so how much methane was either being taken up by the microbes in the soil or produced. So the negative would be an uptake of methane into the soils, and a positive value would be a production of methane. We also collected soil moisture, soil temperature, and conductivity data. You'll notice that I've described these as mean values. This is because each of these is actually a mean of 6 samples. So over a 3-day period, I went out there and collected data in the morning and afternoon, and all 6 of those samples are summed into summarized into a mean. Do note, if I was to publish this, I would want to also include a measure of spread of the data. This is a measure of central tendency, the mean. I would want to include a measure of the spread of the data, or a measure of dispersion, such as the standard deviation or the standard error. So this is the data that we'll be working with in order to look at box plots, to look at scatter plots, to look at t-tests, and to look at regression analyses.